Hello and welcome to DNA 1301, Introduction to Deoxyribonucleic acid. The topics we'll be covering today will include the DNA structure as well as how it stores our genetic information. We will then move on to debunk the myth that human development is entirely determined by our genes. Let us begin by understanding the rationale for learning these topics. The aim of this video is to facilitate later understanding of more advanced topics relating to the DNA. This includes topics such as DNA replication, gene expression, and mutations. To achieve this goal, this video attempts to help us establish a strong foundational knowledge about the DNA. The second goal is to correct potential misconceptions about genetics, while our understanding of biology is still at its infancy. As previously suggested, this video attempts to do so by correcting a common misunderstanding that our future is entirely determined by the genes we have inherited. Let us now move on to the scientific content proper by first considering the components of our DNA. 4 different kinds of nucleotides can be found in the DNA, and all nucleotides contain a phosphate group, shown in yellow, and a 5-carbon sugar, shown in blue, which is known as deoxyribose. What distinguishes one nucleotide from another is their nitrogen-containing base. These bases are adenine, which is shown in red, thymine, shown in purple, cytosine, shown in orange, and guanine, which is shown in green. Additionally, the nitrogen bases can be further categorized into pyrimidines and purines. Essentially, pyrimidines have bases with only one ring, whereas purines are larger and have bases with 2 rings. Thymine and cytosine are pyrimidines, whereas adenine and guanine are purines. Now you may wonder, how are all these components connected to each other? In the deoxyribose, the carbon atoms are labeled 1', 2', 3', 4', and 5'. All nucleotides have a nitrogen-containing base that binds to the 1' end, and a phosphate group that binds to the 5' end. Through condensation reaction, the OH group in the phosphate group of one nucleotide reacts with the OH group at the 3' end of another nucleotide. This reaction produces one water molecule and establishes a bond between the 2 nucleotides known as a phosphodiester linkage. In a DNA molecule, 2 sugar phosphate backbones are connected to each other through the nitrogen-containing bases. Adenine can only bind with thymine, and such pairs are held together by 2 hydrogen bonds. Cytosine can only bind with guanine, and such pairs are held together by 3 hydrogen bonds. This pairing of bases is referred to as complementary base pairs. Because every complementary base pair contains a large purine and a smaller pyrimidine, a DNA molecule has a uniform width of 2 nanometers. The relative amounts of nitrogen bases follow Chagaff's rule. The first part of the rule states that the proportions of adenine, thymine, cytosine, and guanine are species-specific. The second part states that there are equal amounts of adenine and thymine and equal amounts of cytosine and guanine. Next, we shall consider how a DNA molecule serves its function of containing all our genetic information. The answer lies in a sequence of nucleotides, not the number. A short sequence of 5 nucleotides can yield over 1,000 different sequences. This can be calculated by using the number of nucleotide sequences as the power for the 4 types of nucleotides. Therefore, since the human DNA has billions of nucleotides, it carries a capacity to store an incredible amount of information. It is also good to know that the number of nucleotide sequences in a DNA does not determine the complexity of a species. Now that we know the basic components of a DNA and how they fit together, we are ready to look at the molecule as a whole. Recall that a DNA has 2 sugar phosphate backbones. These resemble the poles of a ladder. The complementary base pairs resemble the rungs and hold the poles together. The 2 chains of nucleotides are also anti-parallel to each other. One strand of the DNA has a 5' end at the top of the ladder and a 3' end at the bottom of the ladder. Another strand of the DNA has a 3' end at the top and a 5' end at the bottom. Now, can you identify the missing feature of this DNA model? Yes, a DNA molecule is not a simple straight ladder. It has a twisted double helix structure. Because DNA encodes our genetic information, it clearly has a role in influencing our development. However, the influence of our genes is not absolute. Human development is the result of gene-environment interactions and is rarely based on genes alone. Let us consider the importance of gene-environment interactions by examining an interdisciplinary study involving both biology and psychology. The study, conducted by Caspi et al., focused on the serotonin transporter gene, and data was collected from participants for 26 years. A serotonin transporter gene can have three possible combinations: a pair of short alleles, a pair of long alleles, or one of each allele. The authors found that the type of serotonin transporter gene alone does not influence the likelihood of an individual being diagnosed with clinical depression. However, compared to people with LL or SL alleles, individuals with the SS alleles are at a higher risk of developing depression when they experience more stressful events. This example shows that our genes hold a substantial influence over our psychology. It also illustrates that psychological development is not entirely determined by our genes. Rather, the environment we live in also moderates our development. We are now prepared to proceed on with more advanced topics about the DNA. As we further our learning of biology, let us not forget that the genes we have inherited are not the only things that determine what we will eventually become. This can be summed up with a quote from Roy T. Burnett: "You are not the victim of the world, but rather the master of your own destiny." With that, I end my presentation. Thank you for your kind attention.